Hello, we're going to do chapter 44, which is osmoregulation and excretion, kind of continuing with the theme of digestion. At the end of digestion, we had excretion of solid waste. So we're going to look at the urinary tract system. So we'll do a little bit of looking at different life forms, fish, birds, and, mammals and all of that stuff. And then also looking at invertebrates, how they maintain osmotic pressure. Stop it, sweetie. Okay. Why do I have this problem every single time? Can you go to the next chapter? Okay. Mainly organisms are divided into two groups, osmoconformers and osmoregulators. Before we talk about what those two big words mean, the word conformer meaning you should know the English meaning of that word, right? They conform to their surroundings. Like one of you. And a regulator is basically they regulate their internal temperature, their internal balances, regardless of what the outside temperature, outside conditions are. For example, we are homeostasis. We have we mammals, us human beings, we have homeostasis. We regulate our internal temperature regardless of what the temperature outside is. Like today, it's 120 degrees. The inside of my body is not 120 degrees, and when I'm in Connecticut and the temperature is like minus 47,000, the inside of my body is not minus 47,000. It never is. That is homeostasis. A part of homeostasis for us is also regulating the amount of water that is in our body, regulating the amount of salt. When through the chapter I'll use the word salt. In this case, I'm not talking about salt as in just NaCl. I'm talking more in terms of ions, whether it's Na plus, K plus, Cl plus 2, idiot. Cl minus 1 and Ca plus 2. It could be any ions. A salt is basically just that, right? Basic chemistry. Salt is ionic bond. It has a positive ion and a negative ion. There's a cation and an anion. When it dissolves in water, almost all, every single ionic compound dissolves in water. So when they dissolve in water, basically it means that the positive ions kind of form this weird nest-like thing around the O minus O. H2O is a polar covalent bond, so O is partially negative and H is partially positive, so it kind of forms that nesting kind of thing that happens. Osmosis. So before we get into all of this, so that's the idea of regulation. We do regulation. We're homeostatic. So why am I messing up that word? It's homeostasis. Took me a second to before that, let's look at what diffusion is. All of you know what diffusion is. Diffusion is when things move along the concentration gradient, which means it goes from a high concentration to a low concentration till they kind of become even. Osmosis is the movement of water from a high concentration to a low concentration. I think in chapter 7, we've talked about isotonic, hypertonic, hypotonic. This is just an extension of that. Osmotic pressure is the minimum amount of pressure required inside. This is taking into consideration inside and the outside of the body. So the minimum amount of pressure required inside the body to make sure that there is not an inward flow of water. So for example, if you take fish, so I think my next slide has to do with fish. So I'm going to put fish aside for a second and I'll come back to it. Plants actually don't have, they don't have any specialized osmoregulation, what do you call them, organs. What they have is a stomata. Stomata is on the underside of the leaf. It regulates the amount of water that comes in and carbon dioxide that goes out, comes in, goes out, oxygen that comes in, goes out, all of that, it regulates all of that. It has these guard cells that open and close, and we'll talk more about that when we do photosynthesis. Whereas in terms of mammals, we have kidneys that do similar osmoregulation. Now going into fish. Again, can't move the slide for some reason. Okay. Osmoconformers. Osmoconformers are the ones that kind of keep their body, they conform to the outside environment. This you see a lot of this in marine organisms. Most marine organisms are stenohaline, meaning stenohaline, meaning they can either live in fresh water or they can live in salt water. They're either marine organisms or they are freshwater organisms. They can't really go back and forth because they have mechanisms within their body that kind of regulate, wrong word, conform to the outside of their environment. An exception to this, which is the euryhaline, is the sockeye salmon. Salmon lives most of its life in the ocean, but then when it's about to spawn, it goes upstream into the freshwater. So sockeye salmon has mechanisms that allow them to move between freshwater and marine. Now for so what is wrong with me? Osmoconformers. There are very few organisms that do this. Examples for that is they're very isotonic. They're isoosmotic with their isoosmotic. I don't care what you use as long as you get the point across. They're with their environment. So let's assume, for whatever reason, for just example purposes, let's assume that the outside has 50% water and 50% solutes. So 50% solvent and 50% solutes. The inside of the organism will also be 50% solute and 50% solvent. Solvent here being water. For example, hagfish, skates, and sharks are good examples of this. They are osmotic. Hagfish is like super interesting because one of the ways it regulates the amount of water and salt in itself is by creating this giant amount of sputum, mucus, spit. It can actually create about a bucket's worth of spit in like an hour. Osmoregulators, someone like us, we're osmoregulators. One of the things we actually tend to do is we expend a lot of energy within our body itself to maintain a conducive environment. For example, if you drink a lot of okay, I'm having like the paralysis of the tongue. If you drink a lot of water, you tend to, it kind of dilutes the salt concentration in your body, so you go to the bathroom a lot more. If you eat a lot of salty food, that means you're intaking a lot of salt into your body, which means you need to up the amount of water. So when you go to the bathroom, it's like very, very little. There is a lab we will be doing. Luckily, we do have the supplies for this lab, so we will be doing this lab, which requires you to pee in a cup. So now coming into fish, which are stenohaline. So let's look at two different types of fishes. The first, stenohaline, meaning they do not cross salinity levels. So a freshwater fish will only live in freshwater. A marine fish will only live in the marine environment. Euryhaline, like the sockeye salmon, will go cross. So let's look at freshwater fish. Freshwater fish, the outside, the salt concentration is much lower than the inside. So what they're going to do is they do need to uptake a lot of ions because they do not get it elsewhere, right? It's not like they can go to Stop and Shop and buy a box of salt. So what they do is they take in a lot of water. In that water is a whole ton of ions that go in. There is an exchange of water, ions coming in, water going out, in and out. Water is absorbed through the skin. So a freshwater fish, so for the time being, let's put everything aside and just look at the skin, okay? The environment outside there's a higher concentration of water on the outside than on the inside. So water is moving into the fish, and the ions, remember again, salt. When I say ions, I mean salt. The concentration of ions is much lower on the outside than on the inside. So the ions are moving outwards. Basic diffusion is happening here. High concentration on the inside, low concentration on the outside. So the ions are moving that way. And high concentration of water on the outside and a low concentration of water on the inside. So water is moving in. So that is this thing is happening constantly. In order to maintain this balance, and in order to maintain the water concentration in here and maintain the salt concentration in here, they do take in a lot of water up through their gills. They keep the salt as they go along, and the pee that comes out has a very, very, very low concentration of of salt. So very low concentration of salt in the pee. So they keep most of the salt because they are losing their salt through diffusion. So they have to constantly keep replenishing it. But they are gaining a lot of water, and the way water has to leave. So they're gaining water in here because the concentration of water is greater on the outside than on the inside. Water starts coming in. So as it comes in, it goes through the system and out in the form of pee. It keeps the salts that is coming in from here because it is losing a lot of salts going that way. So this water is this pee is going to be have a very, very, very low concentration of salts. Now let's, oops. Now let's look at a marine fish. In this case, again, there is a high concentration of salts on the outside and a low concentration of salts on the inside. So salt is going to move in. High concentration of water on the inside and low concentration of water on the outside. So water is moving out. So basically what the fish is going to do, it's going to take a lot of water in. The gills are going to take in the dissolved oxygen that it needs for its system, and it's going to pump out a whole bunch of water out right away, which is the opposite of what's happening here. Its pee, on the other hand, is very concentrated in salts. Remember, the salts are coming in and it's trying to get rid of it. It's trying to keep the water that comes, the little bit of water that's coming in, and whatever water is in its system. So this pee, this excretion system of water is going to have very small amounts of water and large amounts of salt. To conclude, freshwater fish is going to have pretty freshwater pee, and saltwater fish is going to have a highly concentrated salty pee. This image, basically whatever we talked about for the previous image, is the exact same thing, kind of reiterates on that. The Yukon syllabus tends to ask 1 or 2 questions on their final exam on this, so kind of go over the difference of osmoregulation and osmoconformation. That's sockeye salmon. It's euryhaline. Stenohaline is where it lives in one environment. Euryhaline, where it can go from one environment to another. Sockeye salmon is one of those organisms that can navigate between 2 different types of environment. So the amount of water balance, let's take the human being for an example. I mean, the rat is pretty self-explanatory. In terms of human beings, we need about 2.5 milliliters of water every day. That's about 2.5 liters. 2,500 milliliters or 2.5 liters. About 1.5 liters of it is actually ingested. So you drink it in terms of. Okay, so let's look at the water balance, and let's look at human beings for water balance. You need about 2.5 liters per day. About 1.5 liters if it is ingested in terms of liquids. Again, when we talk about liquids, it doesn't mean that it's only water. That is a misconception that people have, that if you drink soda, you're not getting water. You are getting water. It's just that you're getting a higher concentration of salts that your body doesn't need. The rest of it comes from food. Chances of you eating dehydrated prunes every day and you're not 95 years old is pretty low. A portion of it comes from metabolism itself. The way you would break a macromolecule is by adding water to it, hydrolysis. The way you make macromolecules is by doing dehydration. So when the body is making its own proteins, when the body is making its own DNA and stuff like that, a lot of water is released. So that is about 0.25 liters of water is released through metabolism. Remember, metabolism has two functions to it: anabolism and catabolism. Anabolism is when you make something, when you make a larger molecule, when you make large macromolecules using monomers, you are